So hi guys, nice to see you here today, and it's great to be here. Actually, I walked from the company today, so it's 2 kilometers away. And I would like to start, first of all, with some kind of gaining interaction with you. So that's the first slide you can see. And I would love to check your background. So first of all, how are you today all? Fine? Great? Open for gaining new knowledge? Fantastic. Okay, I know that that's the Faculty of Mathematics and a great data science background here behind, so I will ask you something else. So this is the slide that I use to ask different questions. I'm generally asking people if they used AI. And people do not know that they are using AI without their knowledge every day, on everything actually right now. But I will ask you, do you know how the drug development process looks like? You know. So some of you know, some of you don't. For me, it's better that you do not know, that I can share you something new. But I will touch upon drug development process, drug discovery and development, even with the connection to a patient at the last slides. So, as I've been introduced today, I'm Kaja Milanowska-Zabel. I have a PhD in bioinformatics, but my passion is right now business. So MBA here, and I would like to talk about decoding the future. So usage of AI, but from the business perspective, how it brings value in drug discovery process. So I will share with you use cases, how to actually AI can be used in drug discovery process, but, sorry, I will not go here, but in a way, how it brings value to not only drug pharma, drug development companies, but also to patients, and I guess I need to go there. Now that's better, okay. So first of all, starting from drug development process. So I don't know if you know that nowadays it took like almost $3 billion to bring one drug to the market. And looking from the 1970s, it was like a really tiny cost to bring a drug to the market. And even though the cost dramatically raised, it's still like a huge effort to bring one drug to the market. It takes like 10 to 15 years, including all the parts of drug development process, starting from the early drug development, when you can see like 500,000 new potential drug candidates will go through the process to eventually reach only one drug at the end. So it's like, first of all, huge effort, lots of years, high risk, and lots of costs to actually reach to the market. And how the AI can change that? I guess you have your own ideas how it can be used. Of course, it's like making the time shorter and reducing the costs. But in general, the things that can also AI help here, as a tool, of course, it's finding new discoveries that were not accessible till now without the usage of the novel tools. So in general, it is called the intelligent design. So you can say it's like augmented drug design, where AI helps scientists to make informed decisions in which way to go with the usage of AI. So that's something that I would like to share with you today. And if you have any questions, any comments you would like to discuss, please do. Please raise your hands. I want to have the interaction with you today. It will be better for both of us, I think. And now, of course, I'm from Artigen, so I need to show you something about the company today also, but in a way, how you can actually look at the usage of AI in drug development process and how it should be used. So in general, you know that now AI is everywhere. It's like a buzzword. Everyone has AI. Yeah, of course, true. And there are lots of the companies now, like mushrooms after the rain, like popping, you know. You go to the conference. I've been last week at the in Basel at the Drug Discovery conference, and it's like, okay, so how are you different from all of those there? The companies coming from different backgrounds, like finance, e-commerce, they have a great data science behind, but actually they, like the models that are building give like great rock AUCs, fantastic, you know, mathematical results, not actionable insights in biology, really. And what is the most important here? I call it biology-first AI. It's not biology-driven. It's the biology behind. So chemistry, biology, I know that you have bioinformatics in here, and you fully understand that biology is a process not that easily explained with mathematics, physics, and you need to understand that to actually use tools that are now in a proper way. So that's what I call biology-first AI. Then, of course, expertise. You need to have people that are experts in their fields, and I'm always saying that our data scientists and software engineers are infected with biology. You know, they are by osmosis gaining the knowledge that help them build proper tools. And from that, you can have accelerated drug discovery. I would check, yeah. Optimized research, improves R&D efficiency, and of course, what matters the most for all the business behind is increased return on investment. So from that point, we'll today together look how AI can speed up the process and bring value to the end users. So it's the patients at the very end. Of course, pharma, money, time, but at the end, there is the patient that needs that. We are on the market for 10 years already, and we've done a lot of different projects, so I think that's why I can tell you more how it looks from the behind, actually, from the garage point of view, how the research is being done. And we'll start with the data. That's nothing new, of course. We are data scientists. Data is all you care about. Data is the new oil. Data is money. But you need to have a data in a way that can be used in different research approaches to give valuable insights. And what we've seen is that I've talked with the pharma companies that have, you know, tons of data, and they are like, okay, so we generated that data because we thought it might be useful, but we did not know how to use it and what for to use it. So the multi-omics approach right now, of course, if you have the idea how to approach that, and addition of biology-aware AI can improve the process, actually. So if you're a pharma company and you have tons of data, the next thing to do is to find a great data scientist that can use that in a wise manner that will speed up the process. We had projects where we've seen that you can speed up the process from 3 months to 2 days. Of course, cost reduction. In-house process means that there is the time to the market, which matters the most, shortens. And regarding the labor needed, less of the lab work is needed with the usage of, well, let's call it machine learning in general. We do not have a true AI. So with usage of the proper tools. So what kind of data we have in biology, in life sciences? There are different data modalities. So all the kinds of omics data you can imagine of. Omics, transcriptomics, genomics, proteomics, immunomics even, spatial omics, cell painting structures. And from that, a new concept in life sciences, like data products, emerged lately in general to give valuable insights. And what does it mean for us? So when we are working with data, you need to be aware of, so you are also bioinformaticians, so you know, as many of you are there, so many data formats will be there. If we had 10 bioinformaticians, we'll end up with 15 data formats. So the harmonization is a key here. How to get all the data domains in a format that can be used in a single data product. The second thing is different, so data domains, the data modalities. With the data modalities, there are not only like imaging, profiling, structural data, medical records, video data, audio data. You know that there's audio data in Parkinson's disease. Actually, you can work on audio data from Parkinson's patients to say if the disease is progressing or not. So then, adding all the data universe, not only the data you have in-house as a pharma company, but looking into the open space to engage all the other data sets that are there. So you are now super happy, you have your data, but what can you do if there is not enough data around? So what would you do? You have like a biological problem and you have like a really scarce data set. What would you propose? Get more data, so generate it, more costs, put money into laboratories, get more data. Okay, but you know, your competitors are not sleeping, they have the data already. What can you do? LLM revolution. So in general, you can actually use the already existing data to create new data. Of course, it's not going to be the same as the true raw data created in laboratories, but with different already existing data sets, databases, and precise initial investment with foundation models, you can try to build your new data sets quicker, cheaper, and be in the front of your competitors. What really matters here, remember, we are in the world of course, patient needs a new drug right now, like for the yesterday. So time to market means help to the patients, but also money, of course. LLM revolution. And now, so from that's the, you know, the general overview of starting a drug discovery process. Of course, when you are like sitting in your home and thinking, okay, I will start a drug company. First of all, you will need to have the idea in which direction. Second of all, you need to have a data. So we have it. We have data, different data modalities. We have like data universe, multimodal fusion, which is like a hype word right now. Everyone on the conferences, do you do multimodal? It's like, you know, a secret. Can you do it? And multi-domain adapter, and from that you can go into insights, different new hits, so new drug candidates, biomarkers, agent selections, mode of actions, patient stratification, indication expansion. That what it actually goes into drug discovery process. And now we'll dive more into it. So from insights to impact. And we'll start with a traditional drug discovery process. So I will, because you know, I could talk for hours about drugs, life science, but we'll go through like the most popular process. You have in drug discovery, you have two ways you can follow. First way is traditional target-driven drug development process. Second is phenotypic-driven. Target-driven means you have the indication of your choice, some kind of a disease, you are looking for a target that you want to then target with your molecule to heal the patient. And target ID, so identification, you can, of course, now do it both ways. Hire a person that will sit in front of the computer, look from all the information in the internet, PubMed, patents, known targets, and so on, and try to find that. Or you can use data science. Gather all the information and prepare high throughput data for that. Multitask model with feature importance, where you have different vectors with different data encoded, and then do different predictions from that, at once, of course, and stratify the groups in a way that will be connected, for example, the patient subpopulations with given genetic mutations, genetic perturbations, information about the potential target that can be then used in a drug development process. Second of all, there is the target validation. So we've done a project where we had the information from synthetic lethality from different orthogonal studies that we had targets, like molecule target pairs, already validated. And we generated the group of potential new targets that can be a new targets in the given indication, and by retrospective study we looked for groups where from the orthogonal studies the groups were already validated. So actually for that you can then prioritize your targets that will go into the lab validation. So you will not generate use thousands of dollars to do a lab validation, but you will prioritize those in a way that you can choose only top 10 to go to the lab. Cost reduction, time reduction, higher chance of getting the right target quicker. So the general overview regarding the target ID and validation using usage of AI is a risk mitigation. As you can see identification of biomarkers, new targets from given groups that are confirmed from the orthogonal studies. You can get thousands, tens, hundreds of new predictions that will give you the information about the features associated with it, so higher explainability. And you can increase the percentage of tasks that actually improved with the usage of AI compared to the traditional way of working. So now we are super happy. We have our target. So you are already in a moment that, so you started the pharma company, you had the data, you found the target. Yes, fantastic. What to do now with it? So what types of drugs do you know? There are small molecules. Do you know small molecules? Do you know biologics? Biological drugs, antibodies, gene cell therapies? Okay, fantastic. So now you'll need to choose which way you want to go. So which way will you go? Which way do you think is easier? Small molecule or biologics? Much easier, small molecule?
No, how do you define easier?
Is the biologics easier or is
Easier for the patient?
Easier for getting through the clinical studies or getting through the translational studies. Biologics. So in general, right now, there's plenty of small molecules on the market. Not that many biologics yet. The predictions are that 2030 it's going to raise, and I guess 2050 it's going to be half and half. So 50% biologics. Biologics are right now a rather novel class. of treatments, and like for example, in immunology, the checkpoint inhibitors were the first in class treatments like that got to the market like lately, really lately. And so from the pharma point of view and looking at how the process looks like and the money needed, small molecules are rather right now, let's call it easier. But there are more of them, of course. So, yeah, more patented, yes. So let's start with this small molecule discovery. For now, I will like slightly change the point of view of the drug the discovery process, because that's actually a phenotypic-driven drug discovery process. Phenotypic-driven drug discovery process means you have no target. So I will get back to our target in a moment, because you've chosen biologics. So we get back to our target, and we decided to go through biologics. So great, okay. So just to show you how the AI can be used also regarding different approach, so phenotypic-driven drug development process. In that way, so for example, there is the Cell Painting Consortium brought to life by Broad Institute and Ann Carpenter. The idea was to generate thousands of images of different cell lines that are like different indications, different diseases, different states, and then treat those. So make pictures of them, of course, and then treat those with different molecules of known mode of actions. So known chemical structure, known properties, known like physical-chemical properties, known mode of actions, and then check the output of it and make pictures again. And in that way, you could find which small molecules were responsible for changing the phenotype from diseased to the healthy one. Those are called perturbagens. And with that data set and pre-trained models, you can then use those models to find a new perturbagen, a new small molecule, a new lead, so candidate to a lead molecule in unknown data set. And yes?
LLM revolution for generation of new data.
Yes, yes.
Could you use this to generate
Yes,
such
yes.
data? You? Okay, so you can use LLMs to generate these images and Yeah, so my question was just what you heard already, if LLM can be used to generate such a data and use it for further development of the drugs. And so, and you do use this in Artigen.
So, sorry. So actually, yes, we are using, you are using the data that is like from the open source from the client to also indicate new data, so to guess like the new data. You can see the assays there from the cell lines and compounds, and the more lines and columns you have filled, the more you can guess. So you can extrapolate those. So actually you can use that to extrapolate to generate more data out of it. And here, I don't know if that answers your question, but the answer is, in general, when we have, so the cell painting data is not that easy to generate and not that low cost. So we have companies that are like biotech companies and that they have not lots of data. But we have our pre-trained models, we can use their data and we can extrapolate the data into the other types of cell lines or small molecules that then can be used for the usage of the, like, for the sake of the experiment. And in that, here you can see it's like, yes?
It's that extrapolation works. Have you checked how this extrapolation works?
Well, yes, of course. So, like, you know, I'm not, just to tell you, I'm not a data scientist. I'm bioinformatician and business now person. But yes, we've done that. So it's not in a way that, I know that data science team actually building the models did all of those. They of course checked the models, how they worked, and if you cut in half and check with if actually generates the other part in a way it should generate, I know if it's been checked. I do not know, you know, the exact numbers for that, but it was done. And you can see here that in general, to get that the usage of it, you can use the images. You will use then the omics data. Here is RNA-seq, so transcriptomics data, so the expression of the genes, but you can use any other types of omics data, and ligands meaning the structures. And from that, you can get new hits, so hit identification, bioactivity, modes of action, library design, lead optimization. The way from that further would be so-called target deconvolution, so guessing the target out of the molecule, but it's really hard. We are doing that, but it's not that straightforward as a typical target ID. So we'll get back now to the traditional target ID-based drug discovery and biologics. So you said biologics, we are getting into biologics. That's actually a good choice, because the predictions from the market are the biologics will take more of it and are more expensive. So for biologics, how to use then machine learning data science tools in biologics. In drug discovery process, first of all, you need to identify the hit, then you need to prioritize the hits, so hit to lead, and then you need to optimize the lead. So in each of those, I will show it to you on the base of antibodies, because it's like the easier version, but you can use it on peptides, linkers, binders, nanobodies, anything you can imagine of regarding biologics. We are doing also mRNA optimizations, so RNA-based therapies. But for the sake of this lecture, I will use antibodies. So for example, you will start with the hit generation, so de novo lead generation. I will touch upon that on another slide. And so I will, like with the discovery, I will give you more details in a moment. Then hit screening. Hit screening means you need to describe your hits. So predict actually how the hit would work in a real life. So developability, functionality, safety, super important. On target, off target, toxicity, super important. Everyone now asks about the safety predictions, and I'm not meaning like typical ADMET hoax, but here, like, for example, off target toxicity. You do not want your molecule to act anywhere else in your body. And then lead optimization, meaning you need to improve different liabilities in your small molecule. Affinity, specificity. Now everyone is asking also about bispecifics, multispecifics. So antibodies like last year on the PEX conference, so protein engineering conference, the buzzword was bispecifics. Last week, multispecifics. So it's like, you know, and so that's something that you can use also. I will not go into details here. I would like to go into details more here in generating new hits. What you can do when you have a target sequence. So we have our target, we are super happy. and you do not need to have a structure. A few years back, when I was doing my PhD, so let's say it was like really not that long ago, you need to generate a structure, you need to have a known structure out there. Homology modeling, Rosetta at home, I remember setting that at my laptop. I was super happy, you know, Rosetta at home, yes, I'm helping the science. Right now, AlphaFold, that's everything. Physics-based methods together with homology modeling, AlphaFold, let's do it. So you can start with your target sequence. From that, you can do generative AI in the sequence space. So like large language models, NLP here, and get to like guess new antibodies based on that. But what is super interesting is generative AI in 3D space. Unfortunately, I will not tell you how it works because I don't actually know from the technical point of view. But first of all, you are doing the modeling of target structure, of course. You have great tools out there already. And then you are trying to get de novo hits based on epitope design. So you need to describe the target, find the epitopes, and then you are looking for those that would bind. The most easy way is antibody epitoping, so finding unknown antibodies and looking for those that actually can be reused. From that, you have generated candidate antibodies. So that thing I mentioned, you need then to describe those using hit description and hit screening. With those, you go into wet lab validation. So we do it's called AI lab loop. So no one will believe you that you have AI and you will do everything with AI. But if you have AI with wet laboratory and you can do wet AI lab loop, then that's great. Because from the lab, you have the information, what worked, what didn't work. You can retrain your models and get another hit generation. So from our perspective, we've done a project. It took like 2 loops to get a really nice candidates that could go further. From wet lab validation, you are doing also then, if you have your your hits prioritized, you do lead optimization, also lab loop, and you are super happy we have optimized leads before our competitors, of course. And now, AI in preclinical candidate selection, what it gives from the business value. Of course, accelerated hit identification. It's something that is really super interesting and super important. That's something that I've mentioned, shortening the process from 3 months to 2 days. Second of all, reduce bias. So if you are using knowledge-based, biology-aware approaches, then you can get more reliable mode of actions for those. And higher quality leads, so you have the higher chance that your lead will go through the drug discovery process through clinical studies. You remember, 500,000 molecules, 1 at the end, 1. And of course, AI lab loop, something that everyone is asking right now, and everyone is asking, do you have it in-house? Because like one-stop shop, we do not want to use different vendors. We want to have one person there. Now, super happy, we have our lead. Now we need to translate that further. So preclinical studies into clinical studies. It's called translational gap. So how to build a preclinical study in a way that will be then translated into clinical studies. So you know, in clinical studies we are using human. In preclinical studies, what we are using? Yeah. Tissues, cell lines, animals, animal models. humanized mice, chimpanzees. You need to build your preclinical study in a way that you will feel comfortable with that in a further process. So what can you use? Of course, machine learning here. So here is the example when you can get the TCGA omics data and CCLE, it's cancer cell lines encyclopedia, so the atlas of cancer cell lines, and build the models in a way that you can have profile those genetic profiles map on the different cell lines. So for example, we have our target, we know our genetic profile, and we want to design it in a way that we'll know it will pass through preclinical study and will be translated then in the human. So we can design our study in a way it's informed. So thanks to that, you can choose the cell lines that would fit the best to your query. The other example here, it was used with CRC, so gastrointestinal cancer, colon cancer, to then map TCGA, CRC. So for example, you have here the UMAP versions, visualizations of the clustering that you can then use given cell line in this oncological indication. So that's when you can use your data scientists to plan the study. So lots of now, like I have 3 slides that will show you why it is so important. Like it's super important right now. We are getting through preclinical studies into clinical studies. You need to do all the regulatory things right now before getting into clinical study. END filling, safety, GMP processes, everything that goes into clinical study. It's not like, you know, everyone said, oh, okay, so it's like with the COVID vaccine. It went like that and no one looked at it. No, it's like so regulated that you really, it's super important right now. You can lose everything in that process here. So higher confidence in research, of course. So you are choosing the right cell lines that you will use in the further process. Second of all, of course, R&D costs reduced. Then informed decisions. So the process is not chaotic. I don't know. You're a scientist, so you know how sometimes research can be chaotic. And so faster go, no-go decisions really is super important. You know that there is like things in business called, of course, sunken costs, so the costs that you already used to get into the project, but also there are costs associated with ending the project. The further you go, the costs to close, so like close costs are higher. So the earlier you will see that that's no-go road, the less you will lose at it. Efficient resource allocation is something like super obvious. So your people can because you need to think of AI. Everyone is like, okay, so AI will steal our jobs. Let's think about it in a way like with a hammer. It's a tool. It can be used in a proper way or not. Hammer will not steal our jobs. AI is, of course, will not. I'm not like a forensic scientist or like the futurologist, how it's called. But right now, it's a tool. So it should be used wisely, and then people can get into things that the AI will not help. And then unlocking value in the data. The thing I've mentioned to you, it's like, of course, it's super obvious, looking at new traits in the data that the human eye will not see. But also using the past data sources. So like those pharma I've mentioned, I really talked to the people in the pharma that are like, okay, so like 20 years ago we generated that data. We thought it would be useful. We do not know how to use it. It's also generated with other methodologies, like 20 years old, and so on and so on. We can now use that data. So that's super important. And of course, also proprietary intelligence, meaning super important, like biomarkers for the patient stratification. We are now getting into clinical studies. We need to choose proper patients, so recruit proper patients to our clinical study. Super important. Yay, we managed to get into clinical study, phase 1. Yes, we can now do a publication. Some, press release. We are there. Few more years, and you will get the treatment. But no, that's super important phase right now. We are getting into clinical studies, and there are plenty of examples that can be used here using the AI. Or maybe let's think of those. How would you use now machine learning in clinical studies? What can be done with AI? Any thoughts? Maybe clinical study design. You can design a clinical study. Like patient recruitment. So stratification of patients with biomarkers to get the proper people into our study. Any ideas in that way? Maybe we can
measurements, the tumors, classify the sizes.
So we can do that for the classification of actually, with the design of the study, what would be important to classify during the clinical study to make it successful. Of course, we can use it in image description, definitely. Maybe we can use it in regulatory process. LLMs, you know that there are already LLMs like ChatGPT, like solutions that can give you the template for the FDA or EMA based on the data. So, for example, you can talk with your ChatGPT, Gemini, or however you call it, and hi there, I need regulatory documents for FDA to start a clinical study, and you get your templates for that, what needs to be filled. Let's start with the cohort definition and trial design. So that's something that takes really vast amount of time, and also you need to have knowledge, really great knowledge behind to design a clinical study. So with AI, actually using all the information from the patients, from also for the trial design, you can find, for example, sensitivity biomarkers that will show you what kind of groups you need to recruit to your clinical study. Okay, of course, super important thing is the mode of action. You need to describe the mode of action. So, for example, there is a problem. I don't know if you know the fecal microbiome transplants. So there is something I can talk for ages also about the microbiome, but microbiome is your second genome. And the idea is that microbiome and the composition of microbiota in your gut is connected with the response to a treatment. And there are already treatments that are called fecal transplants. So you can transplant from a donor a microbiome. Although there are also autologous fecal transplants, microbiome fecal transplants, that are used, for example, after treatment of AML. So also the bone marrow transplants that you need to reconstitute your microbiome really quickly to have a really fast recovery. And those are not yet approved by FDA. Why? Because it's super hard to describe the mode of action. How? With, thousands of microbiota they're acting. And then you can stratify the patients in a way that actually the recruitment will be easier. But what is also important here with the trial design, you know that you can look at the clinicaltrials.gov, for example, get the clinical trials that are out there and get the results of those clinical trials and actually look at those that failed. And use that information in your trial design. Why those failed and what to do to have a better design. Second of all, portfolio optimization. Okay, we are going into the clinical trials. We have our small molecule. And when you are designing a clinical trial, it's not always the way that you have the indication in your mind. So, for example, I would like to cure breast cancer, but it occurs that my small molecule can work better in colon. So maybe I will design a clinical study for colon and work further to get that to the breast cancer at the end. So here you can actually use the information, so align drug signatures with different signature of subpopulations and connect molecular profiling with the clinical data to actually choose from your portfolio the molecules that will have higher chances of success in the given trial. Of course, competitors, super important. Always look at your competitors, always. And prioritization of candidate indications for clinical trials. So the other side of that I mentioned before, there you have indications and look for the portfolio, in the portfolio of your small molecules. So just a hint, if you are starting a pharma company, so we started a pharma company, never do one, drug discovery at once. So using AI, you can do plenty of those. And choose different molecules then to go into smaller or large molecules to go into clinical trials, but with your small molecule or large molecule here, you can, as I mentioned, find an indication that it will work better. Also with AI. Super important thing that you mentioned also here, patents. So freedom to operate. You can use data science to look throughout the patentable space and patents databases to find those that are actually patentable. So now, precision trial strategy, how to maximize, yes?
We'll get more mics soon.
Sure. Regarding the previous slide, I wanted to ask, as a person sort of slightly outside of this field, what decides whether something is patentable versus not patentable?
So in general, you need to look at what's actually patented and what are the boundaries of the given patent. And as I remember, you cannot patent the molecule as it is something that belongs to nature, but you can patent the usage of the molecule. So for example, if you have a molecule that was patented in a given indication, even, for example, let's look at breast cancer, you need to look if what it was patented, for example, for HER2 triple negative breast cancer or for something else. That can show you where is your freedom to operate. So like the area of like no man's land. No problem. So now, identify target populations. We know why it is important? Yes, we know what it is. So we can subtype ranking. So every tumor, if you look at the tumor from the biology perspective, every tumor is totally heterogeneous. It has a different core, like nucleus, different surroundings. Like you can look at it as in a forest with, like, different plants, closer to, water, closer to mountains, closer to something else. That's how the tumor looks like. So it's super important to get your subtype here and then identify the optimal cohort. The biomarkers and also IP strategy, so what can be patentable, what not, super important, also mechanism of actions. With that, you will not go further. And also the risk clinical development. So as I mentioned, first of all, informed decisions, but trial simulation. So for example, look at the information from the clinicaltrials.gov, look what failed, why it failed, what can be done different from that. So are we already ready with our drug to go to the market? Do you think? No. Yeah, it passed. It passed clinical trials. You need to make the production. Great. I do not have a production here, actually. But that's super important also. Manufacturing also can be optimized with AI. If you are interested more, just write to me. I can show you materials. Embracing the full process. So something that is also important for the scientists in the pharma companies is how to look for the, maybe it's not important for the scientists, but for CFO, so Chief Financial Officer, definitely. CSO, like PIs, look at the whole process. We've done a project that you can build UI that can be used by biologists in the lab to look at the whole process from the target ID to clinical development and to generate insights automatically, not, look at it one by one, but just to get it there and look at it. And also, there is the super important, like faster turnaround from data collection to actionable results. Something that you mentioned here, that we can go, yes, generate new data. But right now, something that AI can help with here in general in the drug discovery process is to look at the data we have, how to utilize the data, but also get super quick informed decisions if the data was useful or not, or to stop generating that type of data and maybe something more is more important. So it's not like that right now, that we will generate, like, recruit 100 patients and get all the data from them, but we can look at like a group of those, like as a population, and check whether that modality is something we want to collect or not. Really helpful. So there are already, I'm not touching upon any kind of architecture behind that, but like data lakes that can be used with all the surroundings around to look at the data, retrained models, and checking if that data brings new value or not. So that's something that you can use here also to embrace the whole process. And now, so we manufactured the drug. We have that. We are super happy. We can sell the drug. And drug to the market. Something, and now we'll talk about the patient. So pharma already did the money. Now the patient is important. Of course, the patient is important throughout the whole process, but now it's like, the end user of the drug. How do you think AI can help here? Precision medicine is for the target discovery, so rather choose the right, you can choose like something what's out there actually for that patient, but we are not there yet. Pricing, marketing, of course, commercials, yes?
Side effects that you
Also, that's super important. So gathering real world evidence data, super important. So side effects, how to improve the drug, get it quicker out of the market if it fails. But something that I really like, I will share it with you right now. Communication to the patient. You know that there is a problem when you go to the doctor and the doctor says all that medical terms to you and you do not know if you need a lawyer or a translator. So that is how you can, maybe it's too small, but it's like the ECG showed evidence of a prior myocardial infarction. Do you know what that means? So it means that we can see at the image of your heart that you had in the past heart attack. Oh, now I then said, okay, I had it. What can be done about it? Will I have it again? Or so, you know. Super important. And we are actually doing a project like that right now, that we have a pharma company that wants to have that infographics for the patients, design the leaflets for the patient to use it properly. So they are actually caring about the patient, that the patient will know how to use the drug, how it can affect them, what are the side effects of the drug, for example, and not in medical terms, actually. So in general, what is it there for me? And that's something that is more and more now used. We can use LLMs for that, but also VLMs, so visual language models, to make that beautiful infographics, for example. And not only to design, you know, the nice package of the drug, but I think that companies are also, from the marketing point of view, thinking about that, how to, you know, visualize my drug in the pharmacy. So super important, the motivation. Poor understanding is poor adherence and worse outcomes. So in general, if the patient is not using our drug properly, so reports side effects, it also has an effect on our drug in general at the end. And manual translations, of course, for translations for the different markets. We know we want to reach the world with the drug, so it's of course costly. Money is everywhere here. Poor patient understanding. It's both from the point of the patient, that the patient is lost in the doctor's office. But on the other hand, doctors are also like, you know, feeling that kind of irritation when they are prescribing a medicine that will help the patient, and the patient comes after 2 weeks, 2 weeks, and says it didn't help because they were not using it properly. So, you know, super important from the point of view, from the social effect here also. Improved health, of course, increased efficiency, reduced costs, and everyone is happy and smiling, so we have a better world thanks to that. I can go on and on with diagnostics and so on, but we'll now summarize, so go to summary here. And I will ask you, what challenges can you see here? And ideas? Bias. Great. Evaluations of the outcomes.
Yeah, for example, like the simple data that you were mentioning, generating with LLMs, like how do you assess that it's actually like good data?
Okay. Any other ideas?
Still, because when you have lab in the loop, you have the little time required, you still have the lab bottleneck, the web work.
So also the automation of robotics in the lab, helping the lab, like faster turnarounds in the lab work. Let's see. Data quality and accessibility, of course. So not only we all know we need like standardized, high quality data generated by everyone with the same methods and so on, blah, 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 blah. But it will not happen. So that's the challenge, how to harmonize the data, how to generate new data, how to check if the data, like synthetic generation of data worked, and so on, so on. Interpretability. That's something like, so RDGen operates on the market for 10 years already. And in 2000, I've joined the RDGen in 2016 when it was 3 months old. And you know, Recursion, Atomwise started their companies in 2012. So it was the first entrance of AI into drug discovery, into life sciences. And we are not telling back then, so we are of course not using AI back then, like the word, we are using data science and machine learning. And everyone was like, okay, but that's the black box. So I don't know what happens there. Like with neural networks afterwards, with deep learning, it was even more black boxed in general. And people are still afraid of that. So when I'm going to the conferences, everyone is like, okay, but is that explainable somehow? So that's another challenge. So addressing the black box nature, also for regulatory. So there is still a regulation process not being covered regarding the AI-driven drugs. So that's super important. And for the people to actually adopt to that. Generalization and bias mentioned. So of course it's super important. Generalization is like the holy grail. So it worked at my cohort, but will it work in I know in Asia? Will it work in United States? We had a project here. It was done with the American company BioCollective that was founded by Marsha Carlin, whose husband has Parkinson's disease. So she started a company to help him recover. And she decided to use microbiome there. And we've done a project, I can tell about it because there's a press release, so nothing confidential. So in general, they wanted us to find early diagnostic biomarker regarding the microbiome. And they had the US data, only US cohort. And with the microbiome, it changes throughout the time zones, from person to person, super like not unique, super unique, super unique. And so we found out the cohorts from Europe and from Asia to use that in the study also, to make it more generalizable. But still, really, tough task to tackle. Collaboration, so that's something that I really like. You need to speak the client's language. So if clients come, so you have your pharma company, and now you need to know how to talk to people. And we have bioinformatics company, and we need to know how to talk to you. And you know that that pipeline in bioinformatics means something different than in life science. So when we are telling, like, we have lots of pipelines in-house, they are like, oh, wow, so how many drugs in clinical studies? So if you want to have results here, you need to speak the same language, of course. And regulatory landscape, AI is developing so fast that you know how law looks like. It's like the iceberg moving. And AI, So super hard. Now let's imagine the future. Any ideas about the future? Drug discovery. Personalized medicine. You go to the doctor's office, taking. Have you seen the Dropout? So look at it. The Theranos company, So you are going to the doctor's office. Doctor is taking, like physician is taking your like one blood sample attached to your computer, and then you should take that, and that, and you have a depression. You know, so personalized medicine. And other ideas. For like a few years ago, before COVID, it was like telemedicine or something. Okay, but we'll look at from the point of view of data science. Advanced generative models. So it means, as I mentioned, beyond single property. So multi-specific, multi-modal fusions, multi-modal actions also. So the models, generative models that will go to design for the complex molecules and further into biological systems. Deep multimodal data integration. It means that here we had mentioned the side effects. So integrating real-world evidence data into drug discovery process. Enhance explainable AI. So, you know, right now we can tell, there's some explainability behind our models, but we want to know what actually happened. So if you're talking to like ChatGPT, Gemini or something, you can look and ask for them to show the reasoning. So we want that in drug discovery. We want to see the reasoning behind the models. And I think it will be there. In silico trials and digital twins, there are already companies that are doing the digital twin means an synthetic patient in general. So more realistic and predictive simulations of drug behavior. We can do that, but it's still, you know, not that sophisticated right now. It's really simple. And AI-guided automation, someone mentioned here we had a laboratory work. So we are already going there. So biopharma companies are already doing that automation in the lab. They call it like not semi-automation, but rather augmented automation, because they do not believe still in something that is fully driven by machine learning models. So they need to have that points of interaction, checkpoints, that the human person can actually go there and check that. So that's actually something that is already happening, and it will evolve. Key takeaways. We all know, so like talking about obvious things. AI is transforming drug discovery and development. I think you agree with me right now, yes. Accelerates timelines and reduces costs. Super important, business takeaway, use AI to reduce the cost. Of course, in a wise way, because if you use it in a wrong way, that it will not reduce your costs and timeline. You can enhance research and clinical outcomes. And the most important, if you want to use it in a given domain, it does not apply only to biology, but biology is something like harder than like finance, commerce. You need to use tools in domain-aware, domain-first approach. I have the last slide for you. Now forget about it. Forget about everything I told you. So it is all about the relationship. So I'm a now super business-oriented person. So forget about all AI complex algorithm. It really comes to the human connection. Remember about it. All the innovation, all the success comes from the relationship, from the coffee machines, from the discussions, from meeting with the people like right now, like here. I could be joining online, you know. What would be the experience for you? So relationship first, you can build your company, and then use AI there. Thank you. Any questions at the end?
So you sort of gave us an overview of all of those different stages and where and how AI gets applied in this process of drug discovery. Regarding your own company, or sort of in general, how does this commercial field looks like? Do companies like yours, for example, sort of pick a specific single application of AI in this entire pipeline, or do you sort of try to maintain an overview of the entire pipeline and sort of opportunistically, if you have some expertise or you say like in research, you notice some like opening for contribution or sort of business niche or something, then you sort of jump in, or is it more like of a consulting nature? Because I think you brought it up several times that, oh, we worked with this pharma company, with that pharma company. Yeah, I would like to understand a bit better sort of how this field operates in this respect. Because I can imagine that running a big pharma company is very, very big, so it's kind of hard to do everything.
That's actually a great question. So in general, that's true. You cannot like work with everything in the process. So we called ourselves AI CRO, so Contract Research Organization, but we use AI in drug discovery and development. So, for example, there are different fields in the pharma that you need to touch upon, like for that automatization of the robotics of the lab, regulatory processes, setting up a clinical studies, not from the point of view of clinical trial design, but, for example, from the biostatistics on all the regulatory process behind that. So there are different approaches. We are a CRO, so like you can imagine a CRO, so but in silico space. A CRO works in a different fields, giving services, so consultants in general, in different fields. We have 160 people right now with different expertise, so we cover the whole drug discovery and development process, touching only in some points manufacturing and diagnostics. But it really is a huge landscape. So we have companies that are doing only, like, for example, antibody design. So having AI methods only for antibodies. Or they are doing, like we have a Phenate platform that I've shown for phenotypic driven drug discovery. There are companies that have only that. And they are licensing out the technology. We have also our technologies. We are licensing out some of them, but only when they are repeatable. You can, of course, look for a niche that is not yet fulfilled. In biologics, lots of things are not yet touched with the magic touch of AI. So really all kinds of business models, starting from service-based through like license-based, success fee-based models, looking from the point of view not of a pharma company, but like service technology provider. Everything is there, actually. So small, big companies, CROs adding AI parts to already existing laboratories, everything.
The hierarchy are there, so to say? Are your customers the big pharma companies that I can sort of name, like, I don't know, Novartis and so on? Or does it sort of happen hierarchically where like the big pharma contracts someone and then they realize they need your services?
Okay. So we are working actually with all of the different companies. So the ones I can name because I have testimonials from them, from big pharmas, Boehringer Ingelheim. We were working exactly directly with the director of computational team. Genomics England, another one that I can name. That's the, I don't know if you know them, that's a public company in UK. You know that every newborn is now being sequenced in UK? So they have the cancer, rare diseases, and infant right now. And we are there working not directly with them, but through other company. We are working with smaller biotechs, something I can name also, like, for example, Alchema, working in like Alzheimer's disease. We are working with research institutes like Broad Institute, mentioned earlier. And we are working also with CDMOs, CROs. So any kind of that business approach there. Of course, the best way is to get into the pharma company and work with the pharma, but it's also the hardest one.
Thank you for your talk. You showed us at the very beginning that it became so much more difficult to bring drugs to the market, probably also because regulations became more harsher and selective. So I was wondering, what percentage do you think of the drugs we use nowadays would have been approved today?
Great question. So you know that there are drugs that are being removed also from the market right now, regarding all those regulations, side effects, all the information that is being known, all the also trials in mainly in the United States. So I don't know, like, if I should guess, like, the percentage. So in general, why so many drugs still fail? It's, first of all, the regulations being higher standards, but also there are already lots of drugs being approved. So it's not that easy to get into the niches that were easier, actually, to get there before. So now the indications that are left are those harder ones. So harder to actually target with new drugs. I don't really know how many drugs would go through approval right now, but it's definitely highly regulated, and all the outcomes coming from, like, side effects reported, real-world evidence make it harder and harder. But we can just do a guessing game. How many would go through that process? And if you read, like, for example, paracetamol or ibuprofen leaflet, how many side effects? It's like you can die out of it. So don't really know. We can just guess.
We still have a lot of time for questions. So would anybody want to ask something? Yeah, you will join us for lunch later, because there will be lunch break right now. Okay, so okay, I have one more question about because you mentioned first of all, you mentioned that, you know, data is the new oil. Often people say that data is kind of the moat of different startups, of different companies, the unique data that they possess. On the other hand, we all like LLMs, we all like open data, we all benefit from it. And we kind of need more and newer data to advance our science. So I would like to ask you, what's the sweet spot between sharing and keeping the data to yourself? What's the sweet spot for a company like yours?
Well, in general, there are different initiatives already to open the data. So, for example, like the Cell Painting Consortium, we are the technological partner there. It was brought to life by Anne Carpenter, as I mentioned, but I don't know how many, 14 companies, pharma companies joined to generate the data. And it was done in a way that the data is now open. So it was closed for a year for all the people that brought money to the data to actually have that, you know, kind of that long start to be in front of the others, and then the data was opened. I've been at the conference AI in DD a few times. That there are the companies like Roche, Novartis. They are doing their own consortiums, trying to open the data that are in pharma also. So actually not keeping the data in-house, but making it go live. And so the sweet spot for us is, of course, to get the open data from everything. But I guess there is that idea to actually keep the data for some time closed for the companies that actually use the money to generate the data and then make it open. And you know what? So for example, looking at different things that are actually happening right now. If you open the data, you have like tons of people that get access to it, and they can do analysis for free for you. And show some kind of, so the people are like eager to actually dig into the data, publish it on, I don't know, Twitter or some X, sorry, or something. So I'm in the past, you know. So and we can already see that there are those also open initiatives that the pharmas are releasing parts of the data and making people join the competition to make some kind of the analysis of the data. Of course, they are not releasing all of the data, but some part, and the people for free are showing them the traits to follow. So I think that in general, from the like open data initiatives, all of us could actually benefit. I would love to have all the data open, but I know it's not doable. Because of course it's also the patient's data and all the regulations behind it. But in general, the initiatives like that CP consortium, there's now OASIS consortium being settled up, that the data is closed for like 1 year and then it can go open.
Okay, oh, we have one more question.
Hello. Thank you for the talk, first of all. Really nice overview of the whole drug discovery process. And I was kind of wondering, maybe you have some insight. So the sort of the current field is moving towards bigger and bigger models, which means, of course, more data, which publicly might be hard to get, but also more and more compute, which is expensive, which is often hard to get in like public institutions like universities and so on. Do you have any insight? What do you think should new researchers, PhD students be focusing on in this sort of landscape where competing with companies who are producing these large models becomes harder and harder? Like, where do you see the benefits coming from these public institutions like universities, like research groups and so on?
Expertise, definitely. We have in the company, for example, like cheminformaticians, 2 of them, and they are needed. Like from the immunologist's point of view, when I am talking to the companies, like on the conferences, and I'm saying, okay, so we have biologists, immunologists, molecular biologists in-house, they are like, wow, super, great, I want to talk to you. So if you're a data scientist and you are wanting to get in a place that you can have like your unique selling points of yourself, it's definitely getting in the life science, for example, some kind of expertise, like immunology knowledge. Those people are really valued, really like highly valued, because you can build all the language models of like the whole world, but if you do not know how to interpret the data actually after that and make out of it any actions, what for the model is now. So definitely for the universities, for the institutions, expertise is super needed, and focusing on adding to the general knowledge that expertise knowledge in different fields. That's something we are looking for also, definitely.
Thank you. I think we have time for one more question.
Hello. Yeah, thanks for the talk. It was great. I wanted to ask you, you kind of alluded to it in your talk. You were talking about like the biology first AI approach. And it's kind of a tricky question to answer. But, you know, like with developing all of the AI methods, especially for life sciences, sure, it's possible. And I think this is like where the future will go. But for right now, when you're doing all of these things, how do you assess the actual like impact of AI? Like I know that the promise is, you know, like it'll reduce costs, it will like shorten the timelines that it takes you. But imagine that you did like the exact same pipeline that you're doing with AI, but without AI, but with the same expertise, with the same people, the same team. Do you think it would be that much slower? Like, you know, how do you like imagine extracting away the AI? Is your AI actually helping? How do you measure that?
So it's actually with the information from the clients that we are doing the projects with. So as I mentioned, from 3 months to 2 days, it's not something we come upon, but it was the information from the clients. So they were doing that exact same thing with their people in-house and came to us. With one of the companies, we did projects to identify the heat, and we raised the heat ID rate from 5% to 40%. And it was the information from the client. So actually the validation, super important, something we always want to have when I'm talking with the client and signing a deal. I'm always looking for, you know, that part when I can ask the client to validate that. And so always looking also for the laboratory validations to see the impact of AI. Because those companies coming to us, well, of course we have the companies having new ideas. But in general, if you are working with a big pharma, they've already tested different things. So I have a great example that I think I have like 2 minutes to talk about. I didn't touch upon it. So we had a project where the pharma, so it was not the pharma company, it was a small biotech company with a great people, really like, you know, great minds came to us, and they had a small molecule. And the small molecule had the activity needed, but also had off-target toxicity. And they were playing around with the molecule like for 4 years, and they didn't know how to get rid of the off-target toxicity. And when you are doing the lead optimization with small molecule, in general, you are keeping the backbone and playing around with the side bones to change the conformations, to change the additives, and so on. And we were like, our data scientists, it were like, okay, we have that, you know, crazy idea. Maybe we'll change the backbone. And they were like, oh, why? What the? So in lab, you know what that means, changing the backbone. It's changing the chemical, actually, space into the different one. But with AI, when you have all the small molecules, you can do the virtual derivatives. So you then have like 10 to 9 chemical space that you can search through. And we've done that. We've built the chemical space, we did the virtual screening, and we found a molecule for a different chemical group that had the activity and had no off-target toxicity. So that's how it shows, actually, how AI can, in real life, help. And they moved forward with the drug.
So, with that great success story, please give Kaja a warm round of applause.